Hi students, today the topic is applications of molecular techniques like PCR, recombinant DNA technology and blotting techniques. Okay, blotting technique means DNA hybridization technique. You can identify a specific segment of DNA in the whole genome. We have three types: southern blotting, northern blotting, western blotting. Southern blotting is for to identify the DNA fragment, northern blotting for RNA segment, western blotting for to identify the proteins. Okay, useful for the identification or mutation of sickle cell anemia, cystic fibrosis, and phenylketonuria. Second one is PCR. It is an in vitro technique, that is DNA amplification procedure, which produces million number of copies of your desired gene. Coming to the applications, number one is diagnosis. AIDS, tuberculosis, COVID virus, cytomegalovirus, human papilloma virus. All the viruses can be identified or diagnosed by PCR technique. Second one is all the genetic diseases and inherited disorders are diagnosed by PCR. In the court of law, it will be useful, very much useful. That is DNA fingerprinting, which is used for to identify the thieves and culprits, which is used for the parental dispute also. DNA profiling, DNA fingerprinting, DNA profiling are the two very important words for PCR applications. Third one is recombinant DNA technology, that is otherwise called as genetic engineering. That is when a gene of one species is transferred to another living organism. Here the applications are: the first one is agriculture. Genetically modified crops we can uh, generate, and which yield, that is high yield crops. These are all the advantages for recombinant technology. Second one is diagnosis of diseases. Through this rDNA technology, we can identify HIV, AIDS, and mutation also like sickle cell anemia. Treatment of diseases. Number one is genetic diseases like SCID, severe combined immunodeficiency, gene therapy, and sometimes and cholera vaccine, rabies vaccine, ARV, tuberculosis vaccine, typhoid vaccine, hepatitis B vaccine. Then biosynthesis, that is insulin growth hormone and interferon also produced by this rDNA technique. Then antenatal diagnosis like phenylketonuria and cystic fibrosis also diagnosed by